So the PI3K isoform inhibitors are an important class of drugs in CLL. Specifically, the delta isoform is the most important isoform biologically, we believe, based on in vitro studies in CLL. So the first-in-class inhibitor, idelalisib, as you know, has been approved for the treatment of relapsed refractory CLL in combination with rituximab, and it is quite efficacious and in fact shows similar outcomes in 17p deleted patients and not 17p deleted patients, which is excellent. But there's been trouble with some of the toxicities, which our group has shown are autoimmune, colitis, hepatitis, and pneumonitis. And we found that those toxicities tend to be worse in previously untreated patients, as well as those who are younger. We've also found an association with those that have mutated IGVH. And if these toxicities do occur, it's very important to hold the drug at the first sign, because that's the first prerequisite for improvement of the toxicities, and then rule out infection. Sometimes steroids are then required to manage it. Many patients can go back on drug, and depending on what the toxicity was, it may or may not recur. The hepatitis often may not recur. Colitis tends to recur unless patients are maintained, for example, on low doses of non-resorbable budesonide. And so there are follow-on drugs, not yet FDA approved, but a couple in registration trials. One is duvelisib. Duvelisib is interesting because in addition to inhibiting delta, it also inhibits the gamma isoform of PI3 kinase. And it's possible that inhibiting gamma could enhance efficacy in CLL because there is some gamma expressed in CLL cells. But it's also possible that it may alter the toxicity. And that could actually go one of two ways, because gamma is important for T cell and neutrophil function, so that could enhance some infectious toxicity. But at the same time, gamma is important for T cell effectors, and so if those are impaired, that may reduce the autoimmune toxicity. It's a little bit hard to know how that will play out. And in the phase 1 studies in CLL, the recommended phase 2 dose that was chosen based on nice PK and PD data in vivo was 25 milligrams twice per day with duvelisib. And the results look quite good with an overall IWCLL response rate of about 60%, again, regardless of 17p deletion. And the median PFS not yet reached at the last report, with 59% of patients progression-free at 2 years. So quite nice, and we're awaiting the results of the randomized registration trial of duvelisib versus ofatumumab in relapsed CLL, which is expected to read out later this year. The other drug, which is a little bit further behind, which is coming along, is called TGR-1202, and that's been built as a next-generation delta inhibitor because it has rather a different structure than duvelisib and idelalisib. It did take an extensive phase 1 study, a lot of dose escalation and reformulation to achieve clinical efficacy in patients. And we haven't seen any PD data from patients showing that the delta isoform is actually being inhibited. Interestingly, the drug also inhibits another enzyme called casein kinase 1 epsilon, and that has recently been published. And the implications of this in CLL are not clear. Although at this meeting, we've actually seen a poster demonstrating that inhibition of casein kinase 1 delta and epsilon may actually have therapeutic efficacy in CLL. So I think it's a bit unclear how TGR-1202 is necessarily working, whether it's a delta inhibitor versus a casein kinase inhibitor. And the reason this is of interest is because it does actually show a different side effect profile. The adverse events are less, and we're not seeing the typical, what we believe to be on-target delta effects with TGR. And so the question is that because it's not primarily a delta inhibitor or not reaching the target in vivo, because we haven't seen that data, or is the effect somehow being modified by the addition of the casein kinase component? And so the clinical data that have been reported so far with the single agent have been in relatively few CLL patients with relatively short follow-up, and so I'm looking forward to seeing longer follow-up of the single agent data as well as the registration trials that they've initiated. So there are many combination trials with TGR-1202. The company has their own anti-CD20 antibody, ublituximab, which they're combining with it, which looks interesting. And our group has actually been leading a combination study with ibrutinib, in which the results look quite good, and we presented that data at this meeting as well. Because ibrutinib is so effective, it's hard to tell what the relative contribution to efficacy is, but the tolerability of the combination has certainly been good. And I think that potential dual BCR inhibition is a nice mechanism to move forward. And one needs to have a reasonable toxicity profile to do that.